introduction and for the kind words. Good morning, everybody. Happy to be here, part of Nutrition Sciences Seminar Series. So I'd like to talk to you today about some of the research work we are doing in my lab, essentially, that focuses on the application of nanotechnology in food safety. I gave this title, but then I was, a bit apprehensive that I should have probably given a more simpler title, especially because I always defer to Yang Chao for his nanotechnology expertise, and I want to stay on the side of food safety. Nevertheless, we'll talk some nice food safety stuff today that focuses or uses nanotechnology as a tool. So since this is my first time presenting in nutrition sciences, I'd like to give a brief overview of my talk today. Since this is my first time presenting with nutrition sciences, I'd like to begin with a couple of minutes of my personal and educational background in addition to what Yang Chao nicely introduced. I'll follow that up with my current research, some of our research philosophy in the lab at this point, at least, some of our findings, some of our future research ideas, collaboration opportunities, and also we'll take questions in the end, or if you have questions while I'm presenting, I'll be happy to address those. I'll honor the way you guys follow in nutrition sciences. All right, so I grew up in a small town in northern India known as Mathura. It is probably a 3-hour drive from the national capital, New Delhi, but it is only 30 minutes from Taj Mahal. So next time, if any of you are traveling to India and plan to see Taj Mahal, just shoot me an email. I'll probably connect you to some nice places and nice people there who can help you out. The town where I grew up in, it's a suburban town, and the community there goes very hand in glove with animal population and animal science and agriculture. A very high cattle population and a lot of mom and pop shops focusing on dairy-based products. Visuals like this is not uncommon there. When I was growing up, we had regular interactions with animal population, especially dairy cattle. And that sort of started my interest in veterinary science. Therefore, after my high school and undergrads, I joined my DVM program in 2002 in a really beautiful southern city in India, close to the beach. After my DVM program, I did a master's in veterinary microbiology. I wanted to work more closely with the veterinary pathogens. I focused on studying the effects of capsule or polysaccharides in Staphylococcus aureus, which is a common foodborne pathogen that causes mastitis in cattle, and see how the differences in capsule polysaccharides affects the potential of Staphylococcus to cause mastitis. After my master's, I was more interested to work at the interface of human health and animal health, and therefore I joined UConn to pursue my PhD. My PhD research focused on developing strategies to control Listeria monocytogenesis, one of the major foodborne pathogens, has a really high case fatality rate among foodborne pathogens. Really interesting pathogen to work with. After a brief postdoc at UConn, as Yang Chao mentioned, I moved to University of Arkansas. For those if there are grad students in this seminar, if you're interested to learn more about poultry science, poultry safety, or poultry nutrition, that's a really nice school to go to. After my postdoc there, I joined UConn back in 2019. And since then, I've been developing a research program that focuses on food safety. My split at UConn is at this point 70% research and 30% teaching. So the focus of our lab at this point is to develop intervention strategies for controlling foodborne pathogens, both at the pre-harvest as well as at the post-harvest stages. At pre-harvest stages, we are focusing on controlling pathogens in poultry or on produce at the farm level. Whereas in the post-harvest stages, we are developing several strategies where we can use nanotechnology for controlling pathogens on, for example, poultry products or fresh produce as well. And I'll be showing some of the results from these studies as well. Some of the interventions that we are working in the lab right now are the use of plant-based compounds, phytochemicals. These are essentially secondary metabolites produced by plants in response to interactions with the environment, microbes, and so on. We are also using antimicrobial gases, especially ozone. And two of my master students are actually now beginning to investigate the antimicrobial potential of oxygen as well when used as ultrafine bubbles. We are also working with probiotics, or either alone or in combination with phytochemicals. For today's presentation, I'll primarily focus on our results with plant-based compounds. In addition, in collaboration with Yang Chao, we are using nanotechnology as a tool to improve the dispersion or antimicrobial efficacy of plant-based compounds. Some of the foodborne pathogens that we are working in the lab at this point are Listeria monocytogenes. As I mentioned, this is one of the major foodborne pathogens, causes abortion in pregnant women, meningitis in infants and older folks. So very dangerous and interesting to work with. Salmonella and Campylobacter, together these two poultry-associated pathogens, they cause over 2 million illnesses in US every year. One of my grad students, Kim, she's working with E. coli 157, trying to come up with strategies to control E. coli on fresh produce, and she has some really interesting data as well. In the fall, we are planning to start projects with Vibrio, cholerae, as well as some of the hemolyticus strains, and see if we can develop strategies to control Vibrio in oysters and some of the aquaculture environments. So this group needs no introduction to food safety, but I'll take a couple of minutes to just reiterate some of the important points on food safety. And more importantly, I'll share with you some of our research philosophy or rationale that we are using to develop our projects, whether it is for grants or for student projects. So 1 in 6 Americans suffer from foodborne illnesses every year. When put together on a national scale, this translates into 48 million illnesses, over 100,000 hospitalizations, and almost 3,000 deaths. The annual healthcare cost for treating these infections can be over 15 billion USD, and this does not account for loss in work days or associated cost for families. The interaction of foodborne pathogens with their immediate environment, whether it is in the host, for example, in the gut of chickens, broiler chickens, or in the intestinal systems of cattle, in the processing environment, or in food products. The interaction of foodborne pathogens in these immediate environments is very critical and decides whether a foodborne pathogen would persist or perish in these environments. These interactions are primarily mediated by a range of virulence factors and proteins, which help the pathogen to survive and flourish in these environments. And so therefore, one of the philosophy in our lab that we are following is that can we control these virulence factors or expressions such that we can then control pathogens in these environments. Some of the major virulence factors in foodborne pathogens which have been extensively studied are presented here. Of course, this list is not exhaustive. This is just the tip of the iceberg, but I just wanted to touch on some of these. Several of the attachment proteins in foodborne pathogens, such as adhesin pili, adhesins, Vimbriae, they have been well characterized and known to help with survival in the processing environment or even in the gut of foodborne pathogens. Several of the foodborne pathogens produce exotoxins, one of the most popular ones being cholera toxin. C. difficile is an anaerobic organism which will, it's becoming more and more important, especially with nosocomial infections and also in the food safety area. And they produce toxins as well that can cause, illnesses in humans. CDT is a toxin produced by Campylobacter, causes similar, it helps the pathogen survive in the gut environment and also in human gut to cause various diarrhea and enteritis. Similarly, and there are several enzymes, mucinase, I believe, goes along with cholera toxin to cause diarrhea in humans. Flagella help with bacterial motility, biofilm formation, colonization, again, in both animal host as well as in humans. Then capsules and then security systems, especially the ones with salmonella, help with colonization and causing infections in humans as well. So our philosophy is that if we can reduce the expression of these virulence factors, we could potentially control some of these foodborne pathogens. Now, in order to effectively control foodborne pathogens in the food industry, a farm to fork approach is commonly followed. This involves reducing pathogen colonization in the gut of foodborne pathogens. This is extremely important, especially with Salmonella and Campylobacter, because these two pathogens are specialized in colonizing chicken gut in high numbers. The numbers can reach anywhere to 8 to 9 billion per gram of the intestinal contents, and when these contents come in contact with either poultry carcasses or eggs, it leads to product contamination. E. coli 157 is equally important. It colonizes in high number in cattle, especially in the rectoanal junction of beef cattle, leading to carcass contamination there as well. So therefore, the first step towards improving food safety begins with reducing the gut colonization of these pathogens at the farm level. Reducing the persistence in processing environment, whether it is fresh produce washing facilities or packaging houses, is the second step. And this is extremely important with regards to Listeria. The largest foodborne outbreak of any bacterial pathogen in US was in 2011 because of consumption of contaminated cantaloupes which were contaminated with Listeria. And subsequent investigations into this outbreak revealed that the packaging environment where the cantaloupes were processed were contaminated with this pathogen. Another international outbreak that occurred in 2017 in South Africa had similar themes and fingerprints where the environment was contaminated with Listeria. So therefore, developing strategies to improve the cleanliness of processing environment is very important. Of course, if the pathogen is able to jump the first two hurdles, developing strategies to have post-harvest contamination steps is very important, especially with poultry products and fresh produce. And I'll be talking about some of the experiments that we are doing in this area. And lastly, if a pathogen is super strong and is able to jump the first three hurdles, we are interested to develop interventions which we can use at the level of humans, where we can potentially control the pathogen and change the outcome of the disease. So for today's seminar, I'd like to share with you 4 stories or 4 research lines that we have developed in the lab, where we are using plant-based compounds in their nanoemulsion forms for controlling some of these pathogens at each of these 4 stages. So as I mentioned, reducing pathogen colonization in food animals is the first step towards food safety. In this particular study, we investigated the efficacy of a plant-based compound known as trans-animaldehyde for reducing Campylobacter jejuni colonization in broiler chickens. Trans-animaldehyde is obtained from the bark of cinnamon trees. It is a very popular spice, commonly used in, has been approved in food safety, has a GRAS status. I even came across cinnamon flavored peanut butter one time when I was shopping in Price Chopper. So it goes to show how well accepted this particular spice is. So this slide shows the mechanism by which Campylobacter colonizes chickens. Campylobacter is a really hardy pathogen to control, especially because the birds which are colonized by this pathogen show no symptoms at all. So it is very difficult for poultry scientists to discern if a flock is colonized with Campylobacter. The only way to do that is through fecal swabs followed by culturing in the lab. Within the gut of the bird, the pathogen uses motility and its capacity to attach to epithelial cells to really colonize in high numbers. So if we can reduce Campylobacter's capacity to move and its capacity to attach to epithelial cells, we can potentially control its colonization in birds. So using standard motility assay and attachment assay, cell culture-based assays, we investigated if trans-animaldehyde can reduce these 2 virulence traits of Campylobacter. And we were really excited to see that when Campylobacter was exposed to trans-animaldehyde, it reduced its motility by almost 75% as compared to our control. Similar was the finding with our attachment cell culture-based assays, where we saw that when Campylobacter was exposed to really low concentrations of TC at 0.01%, the pathogen's capacity to attach to chicken intestinal cells significantly reduced by almost 2 logs. These were really promising in vitro results. We followed that up with a whole cell proteome analysis of Campylobacter when it was exposed to similar concentration of TC at 0.01%. And our proteome analysis revealed some interesting findings. We saw that a cluster of proteins which have been proven to contribute to gut colonization or survival to acid in the gut, as well as survival to macrophages, were significantly reduced in their expression. So based on our in vitro findings and proteome results, we thought, okay, let's try this out in our actual in vivo bird studies. However, when we administered transaminaldehyde in the feed in concentrations ranging from 0.1, although we tried up to 5%, but then above 1 or 2% was causing body weight losses. However, we did not get consistent results when the essential oil was added in the feed. We had reductions in one trial where we repeated the study, we did not find any reductions. So it was pretty inconsistent. So at that point, we decided to switch our mode of application and try out transaminaldehyde nanoemulsions in water in concentrations, similar to what we tried before, starting from 0.05 to 1%. For majority of the results that I'm showing today, the nanoemulsions that we have tested out are primarily of two kinds. One in which we have used tween AT as an emulsifier, and the other one in which we have used gum arabic or lecithin combinations as emulsifiers. We developed these emulsions in collaboration with Dr. Luo's lab, followed by their characterization for their size, PDI, zero potential, and so on. So here's the design of the study that we used for our bird experiment. We did a short 14-day trial in which we grouped the birds when they arrived on day 0. So the birds were grouped into control. These are birds which are given normal feed and water, so no essential oil treatment. We had a tween control because we were using tween as an emulsifier, and then 2 doses in fact, we had tried a range of concentration, but I'm showing results for only 0.25% for this seminar. So we had a TC treatment and then a transaminaldehyde nanoemulsion treatment. On day 7, we challenged these birds with Campylobacter, followed by necropsy on day 14 to see how well the pathogen has colonized and if the administration of the transaminaldehyde nanoemulsions in fact reduced the colonization, if any. So here are the results for the study. We had 2 trials, and in both trials, we observed that in case of control birds, now these birds are getting normal feed and water, no essential oil, and were challenged with Campylobacter. In line with previous literature, we saw that these birds were heavily colonized with Campylobacter to almost 8 log CFU per ml concentration in both the trials. However, birds which were administered transaminaldehyde in their diet or in their water had significantly lower Campylobacter counts by approximately 1.5 log in trial 1 and around 2 logs in trial 2. Previous literature with Campylobacter shows that a 2 log reduction in birds translates to almost 34 reduction in human infections. So we were really excited with these findings with these 2 trials. Follow-up microbiome analysis revealed that we saw that there was not a major shift in the microbiome of the birds when they were fed with transaminaldehyde nanoemulsions, except for the Clostridia group. And then when I was conducting a literature search to understand what really does this mean by, if we see a shift in Clostridia, I saw that in microbiome studies where we have used Campylobacter vaccine developments, those researchers also found a similar shift in the Clostridia. So probably just it is possible that when Campylobacter leaves this nichate, it is colonized by another resident bacteria that is present there. But we need to dive a bit more deeper into this investigation. Another compound that we explored is eugenol. It is obtained from cloves, again, GRAS status compound, well accepted in the food industry. These findings were interesting because they were slightly different from what we observed with transaminaldehyde. For example, the results here are shown for how eugenol changed the proteome of Campylobacter jejuni. And interestingly, we found that some of the proteins which were not changed by transaminaldehyde were either downregulated by eugenol. So for example, a couple of very important proteins again in Campylobacter, CADF, 4A, they are important for attachment in the intestine. Several of the locomotion proteins were downregulated. We were excited to see that LuxS, this is a very important protein which contributes to quorum sensing in several microbes. This was downregulated as well. We also found some proteins which are upregulated, for example, protein contributing to the multi-drug efflux systems, probably as a response to the presence of eugenol. So based on these findings, we followed it up with an in vivo bird study, and our results were similar to what we saw with transaminaldehyde, where birds which were fed, given eugenol nanoemulsions in their water, had significantly lower Campylobacter counts in their ceca as compared to the control. So again, promising findings. Especially trial 2, we had almost 2.15 log reduction in the pathogen load in the ceca of the birds as compared to controls. This is a recent study from one of my grad students who is doing his PhD through Shan, and he's exploring the potential of transaminaldehyde nanoemulsions against Salmonella enteritis. So this study, I believe they finished a couple of days back, and he wanted me to put this slide in to share with the audience. Here we have, so interesting findings here as well. The tables here show the level of Salmonella in the cecal contents as well as in cloacal swabs. And we saw that birds which were given transaminaldehyde nanoemulsions in their water had significantly lower Salmonella positive samples as compared to their corresponding control. And this was the same was the trend we saw in cloacal swabs as well. Of course, this needs to be explored a little bit more with the repeated experiments, but something which we wanted to share with you today. So based on our findings so far in the last couple of years, some of our future ideas, we are interested to explore combination treatments using multiple phytochemical emulsions, study the effects of these emulsions on the meat quality or performance of birds. We are also interested to see how administration of these nanoemulsions affect the transcriptome of the pathogen when it is inside the gut, host response, as well as microbiome changes. And based on our findings with chickens, we probably will expand these experiments to layer birds as well as turkeys in the future. So I'd like to switch gears and talk about our second line of research experiments, where we are investigating the effects of phytochemical nanoemulsions for controlling the persistence in processing environment. Are there any questions? I can take a couple of questions on the first area of research if you have, before we go into the second session. Okay, we can probably take in the end of the session as well. So as I mentioned, the second area of research that we are developing is exploring the potential of plant-based compounds, the nanoemulsions, for reducing the persistence of foodborne pathogens in the processing environment. And this is extremely important with Listeria. Listeria has a capability to form sanitizer-tolerant bacterial biofilms. And I have a long-standing interest in studying bacterial biofilms. These are amazing microbial communities. So by definition, they are organized microbial systems which are associated with inanimate surfaces, and they produce exopolysaccharide substances over them, which helps them survive sanitizer and various antimicrobials. Fossil records indicate that biofilms have been the norm by which microbes survive over thousands of years. Recent investigations using next generation tools and technology indicate that these bacterial biofilms, which are a community of organisms, essentially behave like almost a multicellular organism where their expression of proteins changes based on their bacterial numbers and how the external environment is changing. More importantly, recent studies, especially the one that I'm interested in, have highlighted that biofilms can also act as a hotspot for antimicrobial resistant gene exchange and also for bacterial evolution. From a food safety perspective, bacterial biofilms are a significant food safety hazard when they are present in the processing environment, and especially when food comes into contact with these biofilms. The image on the bottom shows the various stages of microbial biofilm formation, and I'll take a minute to describe these stages since some of our future results talk about this development. So a bacteria attaches essentially to a plastic or a steel surface, followed by their attachment and microcolony formation. Eventually, these microcolonies grow and form larger mature colonies. These mature colonies then release the microbes that are present within these biofilms, and these bacteria then they find new niche and then develop a new biofilm. And when food comes in contact with these biofilms, then it becomes a significant food safety hazard. And therefore, it is important to control biofilm formation or inactivate mature biofilms, especially where food is present. So in this second line of investigation, we are currently investigating the efficacy of eugenol as well as eugenol nanoemulsions in controlling Listeria biofilms. One of my PhD student, Brinda, she is primarily leading this project, and she has explored the effect of eugenol and eugenol nanoemulsions on biofilm formation, if these treatments can inactivate or kill a mature biofilm, on the production of EPS or exopolysaccharide. As I mentioned before, EPS helps the biofilm survive virus sanitizers. We are also exploring how eDNA production is affected when exposed to these phytochemicals or nanoemulsions. eDNA, recent research has shown that helps with biofilm formation and also with the AMR gene exchange. And she has also studied the expression of proteins in the presence or absence of eugenol and eugenol nanoemulsions. So in our first experiment, we wanted to see if the presence of eugenol nanoemulsions would affect the formation of Listeria biofilms. So in this study, Listeria was allowed to form biofilms on a steel surface at room temperature, 25 degrees centigrade, for 24 hours, either in the presence or absence of eugenol nanoemulsions. And then we compared the bacterial numbers in the control and the biofilms which were exposed to these treatments. So here are the results for this particular study. In case of control, this refers to the biofilm of Listeria which was not exposed to any plant-based treatments. After 24 hours, we had almost 6.5 to 7 log CFU of Listeria present per ml of the biofilm content, which translates to almost 1 million cells. So it has a really high bacterial density. When we expose the biofilms to eugenol in water, no significant reductions were observed. However, eugenol nanoemulsion treatments at 600 and 700 parts per million were able to reduce the bacterial load in this biofilm by almost 2 logs, which translates to almost 99% reduction in bacterial number as compared to the control. We did have a eugenol and ethanol control, and we saw significant reductions here as well. But since ethanol is flammable compound, it is essentially not used for disinfection in an industry setting. So these findings are really promising. Brinda is currently exploring more combinations and more time points in this particular experiment. We followed it up with our investigations on how eugenol nanoemulsions or eugenol affects eDNA production using confocal imaging. Here are the results for the study. This is a control biofilm of Listeria. It has a combination of green, red, and blue dots. The green here indicates live Listeria cells, and the blue indicates extracellular DNA. And previous literature shows that as the level of DNA goes above a certain level in this biofilm, the expression of the cells in the biofilm changes. They behave very differently. Biofilms which were developed in presence of eugenol at 700 and eugenol nanoemulsions had significantly lower bacterial number as well as eDNA content, indicating that the biofilms were reduced in the presence of phytochemicals. Of course, this is not a quantitative study, but something interesting to observe. Following that experiment, we are also investigating if eugenol nanoemulsions can actually kill a mature Listeria biofilm. The previous experiment was more focused on if the presence of eugenol nanoemulsions can reduce formation of biofilm. Here we tested if we can use eugenol nanoemulsions as a disinfectant on a completely formed mature biofilms of Listeria. So the experimental setup was very similar. Listeria was allowed to form biofilm for a period of 24 hours at room temperature, followed by exposure to eugenol nanoemulsions for, we selected a shorter time point of 1 minute, followed by a slightly longer time point with 30 minutes. And after that exposure, we quantified what's the level of Listeria that is still surviving in the biofilm. So here are the results for the study, really promising results here. In case of control, as I shared in the previous slide, after 24 hours, we had almost 7 log CFU of Listeria and the biofilm, so really high bacterial number. Exposure to ethanol or eugenol in water or in ethanol did not reduce a fully formed biofilm, so indicating that once a biofilm is fully formed, it resists the action of antimicrobial pretty strongly. However, when these biofilms were exposed to eugenol nanoemulsions, we observed an almost 5 log reduction in the level of Listeria present in the biofilm. These are really especially at 30 minutes of treatment time. So this is a really promising result, which we plan to work with the industry further to see how we can develop these results to something which the industry can use at their level. Following our results of the inhibition experiments and inactivation experiments, we also conducted a whole cell proteomic analysis to see how cells in the biofilm change their protein expression upon exposure to eugenol. And here are some of the results from the proteome analysis. We identified almost 1,000 proteins in the biofilm. The expression of majority, almost 50% of the proteins was not actually significantly changed. The rest of the proteins, some of them were down-regulated in their expression, and some were upregulated, as seen in this volcano plot. Some of the critical proteins which were down-regulated by eugenol are flagellar hook proteins, internalin A. This is an extremely important protein that helps Listeria to essentially cross the intestinal barrier in humans and also cause biofilm formation. Chemotaxis helps with motility and finding nutrients in bacteria. So we were excited to see that some of these proteins were down-regulated upon exposure to eugenol. Similarly, there were a few proteins which were upregulated by eugenol, such as DNA K and J, probably in response to the presence of the compound. Similarly, there was an upregulated protein, probably because eugenol exerted certain oxidative stress on the pathogen. But overall, really interesting findings in the proteome analysis. Some of the future ideas in this line of research, our long-term goal in this area would be to develop a natural disinfectant, such as a foam-based spray, which can be used at the processing plant level. We are also interested to understand how exposure to plant-based compounds or their nanoemulsion forms affects biofilm physiology. And lastly, I'm also interested to study how exposure to plant-based compounds or their nanoemulsion forms can affect the capacity of a biofilm to exchange AMR determinants, in which case we might explore some of their antibiotic resistant development properties. Moving on to our third story for the day, having effective post-harvest contamination strategies is very important, especially when the pathogens cross the first two barriers and are able to reach our kitchen plates, whether it is salads or cantaloupes and so on. We are conducting a range of investigations in our lab. I'll not be touching on all of them. Probably we'll be selecting few to talk to you today about. These explorations vary from the use of plant-based compounds or their nanoemulsions as antimicrobial washes, fumigation system, antimicrobial coatings, such as on frankfurters, and also as antimicrobial additives, such as in the case of peanut butter. One of my master student, Kim, she is focusing on developing strategies to improve the safety of lettuce and spinach. And another master student who just defended Jody, she explored if we can use plant-based nanoemulsions for improving the safety of poultry carcasses and eggs, especially as egg wash treatments. Washing of eggs and carcasses in the poultry industry is highly recommended, and there are strict laws or recommendations from USDA that are to be followed. Just to give you an example, wash water that is used for washing eggs has to be at least 90°F and at least above 20°F from eggs. Similar recommendations exist for carcass washing as well. And washing of carcasses can range from anywhere from 1 to 8 hours. So based on these findings, Jody explored the potential of phytochemical nanoemulsions in the lab. And we had some really interesting findings. Here are the results from some of the studies that she has done in the lab. This experiment investigated the efficacy of trans-animaldehyde nanoemulsions in inactivating Salmonella enteritidis on shelled eggs, as well as on chicken skin in the presence of organic matter. We had chlorine as an industry control. Essentially, the eggs were inoculated with the pathogen, followed by their wash treatment, and then we calculated what's the level of pathogen that is still surviving. And we had a similar design of experimentation for chicken skin as well. So we had some interesting findings. For example, washing with just, in case of control, that is washing with just normal water, not a lot of pathogen reduction was observed. So if we wash our eggs with just normal water, not a lot of pathogen load will reduce. Same was the case with chicken skin. Washing with chlorine did reduce pathogen load in the absence of organic matter, but the moment we added chicken litter, which is commonly seen in the processing environment, chlorine lost its efficacy and we did not see any reductions. Trans-animaldehyde nanoemulsions were equally effective in the presence of organic matter as well as in the absence. The last bar here shows trans-animaldehyde, and it lost its efficacy in the presence of organic matter as well. Similar trends were seen in case of chicken skin, where TC, for example, was not effective in the presence of organic matter. So was the case, although they are statistically different, but in terms of biologically significant reductions, we saw hardly 0.5 log reduction here. The nanoemulsion, on the other hand, which was prepared with tween, had almost 2 to 0.5 log reduction as compared to its corresponding control. So the take-home message from this slide is that nanoemulsions are effective in the presence as well as in the absence of organic matter, and are a promising candidate for something that the industry can use for either egg washing or carcass washing in the future. Some of the future ideas based on this research are we are exploring the efficacy of plant-based nanoemulsions either alone or in combinations, for example, with modified atmosphere packaging. My master student, Kim, she's exploring the potential of this combination to reduce pathogen load and essentially see how it translates into pathogen reductions, if there's any synergistic efficacy or not. And we are also interested to see if to do further downstream investigation into how pathogens respond when they are present in these food systems based on transcriptomics and proteomic analysis. Moving on to our last story of the day, in which we are interested to develop plant-based treatments, either just plant phytochemicals or their nanoemulsions counterpart for controlling human infections. And this is especially important because if a pathogen is able to jump the first three hurdles, we are not able to reduce in the gut, we are not able to control in the processing environment or on the food products, that then it is very important to have strategies which can be implemented at the level of human gut environment, where we can use certain plant compounds as supplements to control the virulence or the outcome of the disease. The philosophy that we are using in our lab for this area of research is one that of anti-virulence approaches. So in a traditional sense, when we get a foodborne infection such as with Listeria, the normal line of treatment is the use of macrolide antibiotics, which then can disrupt bacterial functions. It does cure the infection, but then it does exert selection pressure. It could modify host microbiome and could lead to development of resistant pathogens, especially in situations where the use of antibiotics is not properly monitored, if there's a misuse, change in the dosage, and so on. In our lab, we are interested to try another approach which focuses on modulating the virulence of a pathogen, so such as the motility, attachment capacity, toxin production, quorum sensing, and so on. The benefit of this approach is that it exerts relatively less selection pressure and potentially would not lead to the development of resistant pathogens. This slide shows the pathophysiology of Listeria infection. Listeria, as I said, is a superbug. It is able to cross the intestine, spread to essentially all organs in humans. If you are pregnant, in case of pregnant women, it can potentially cause abortions, and in infants or older folks, it can cause meningitis. So very important to control Listeria in humans. This slide again shows what happens at the cellular level. Listeria attaches and invades the cells, followed by its spread within the cell. It does not get exposed to the host immune system, leading to spread of the infection and some of the outcomes that we saw, such as meningitis or abortions. So one of the grants that one of the seed grants that Dr. Luo and I wrote and were successful in funding, we are exploring the potential of plant-based compounds for controlling Listeria in a guinea pig model, and which is based on our findings in vitro assays such as these. We observed that when Listeria was exposed to plant compounds such as triacetylamaldehyde, eugenol, and carvacrol, some of their virulent attributes were reduced in expression. So, for example, the capacity to attach to intestinal cells, the capacity to invade, motility, and listeriolecin production. All of these virulent traits were reduced upon exposure to plant-based compounds. We also had, based on our in vitro results, we conducted experiments with Galeria melanella, which is an invertebrate larvae, which can be used as a precursor before we actually move into rodent models or primate models. And in these results also, we observed that larvae which were exposed to plant-based compounds had significantly higher survival rates as compared to the positive control Listeria. So based on these findings, we will be exploring in the fall the potential of phytochemicals using a guinea pig model. We are also interested to characterize how these pathogens respond to phytochemicals in the gut system using either microbiome and transcriptomics approaches. And the long-term aim in this case is to develop a prophylactic or therapeutic recommendations in the future. And this is one area where I'm really looking forward to collaborating with folks in the nutrition science department. You guys know more than me in this particular area, especially with nutritional health for humans. So all feedback and sessions are welcome on this particular area of research. With that, I'd like to take a minute to acknowledge the funding sources. Some of the results that I have presented are supported by USD and NIFA, organic grants, Northeast SERI, Center for Produce Safety, and funding from industry, as well as CNR Hatch and startup funds. This presentation will not be complete if I don't acknowledge my lab group and grad students, Jody, Brinda, Kim, Trushan, Chen, and Atul. These guys are amazing. They pretty much work 24/7 to generate some really high quality data, and they're progressing towards their master's and PhD goals. A quick shout out to proteomics facility and the microbiome facility at UConn to help with our research. Quick shout out to Yang Chao Luo's lab for helping us, many a times with our nanoemulsion characterization. With that, I will probably stop and will be happy to take any questions that you have.
Thank you very much, Dr. Abhi. I heard so many times my names in your presentation.